Okay, so let's look at the start. Let's look at the testes. Let's look at the more close. So again, sperm start in the testes. Now, what we have here, so you have these things called lobules, and they're actually separated by sheets of connective tissue right here. And these lobules, these are contain all these little compartments, and they notice that they all feed toward the same common structure. So seminiferous tubules, you notice that there's these little blobs in the middle of these lobules. So this is just a cross section of this convoluted compact tubules. That's what we're seeing here. That's why it looks like it's separate blobs, but it's actually just cross sections of big snaking structure. So these seminiferous tubules are where sperm are generated, and they start there. Now they end up going into the straight tubules, what we see over here, and it's kind of hard to see in the histology over here. And then we end up in this network called the rete testes. So straight tubules to the rete testes, and then you finally get to the epididymis. And the epididymis, so again, sperm start in the seminiferous tubules, but they're not quite mature, and then they start to mature in the epididymis. Now what we have here, okay, we have a close-up of the seminiferous tubules. Again, we're looking at a cross-section of a lobule of the testes, and we have these seminiferous tubules. So each of these little islands right here, these are a cross-section of a seminiferous tubule. Again, why the seminiferous tubule is actually going in and out of the plane of view of this microscope side. That's why it looks like individual tubes, but it's actually a connective tube per lobule. And what we have here is that the testicular interstitium. Now, in this slide, it looks like empty space. But the thing is that the cells there have, you can notice that there's like little cells right here and these little dark purple blobs. These are nuclei. So even though they're very faint in this picture, there are cells in the interstitium, and this is what we call, again, they're not empty space, but it contains something called interstitial cells or Leydig cells. Now, these are very important cells. Why? Because these Leydig cells or interstitial cells, these produce testosterone and androgens. So these are the primary male sex hormones. Now, what we have here, so why does it look different? What we have here is a picture where it looks kind of, so this is a lumen inside of the seminiferous tubule. And again, the lumen is just the inner space inside of a tube. And what we have here is that you notice that this one's a little lighter, this one's a little darker, right? So what happens is that this is lighter part. It has early spermatids, which are not quite ready to be moved into the straight tubules and rete testes and into the epididymis. But what we have here, now they have sperm. They're ready to move on. This is what it looks darker in this versus this lighter picture. Now here we have cross-section of a seminiferous tubule, so it's a little more showing more of the cells. So think about how sperm is made. You have these cells, and the stem cells of sperm are called spermatogonium. So what you have here is spermatogonium line the outside periphery of a seminiferous tubule, and as sperm mature, they start moving toward the inside of the tubule, toward the lumen. Again, it's a tubule not just in structure, but also in transporting these sperm eventually to to be ejaculated eventually. So spermatogenic cells, these are what generate and develop into sperm. And these are our spermatogonium. Now then what we have here are nurse cells. So nurse cells, these are not actually going to develop into sperm, but they're very important. So they're things that the process of developing sperm requires a lot of nutrients, requires a lot of protection, and also sustaining. So these are sometimes called or the name for these, you might also hear Sertoli cells or sustentacular cells. So they kind of help to sustain the sperm. So again, they don't nurse cells or Sertoli cells do not develop into sperm, but they help to sustain sperm and help them along and grow up into mature sperm. So again, they do not become sperm. They promote the growth of sperm. They are important, but they do not become sperm. And they also form something called the blood-testes barrier. We'll get to that really quickly. All right, so this is looking at further close-up, and now at a cross-section of a seminiferous tubule, but now only a small wedge of it. And what we see is that, okay, but now we have that blood-testes barrier, but now we have all of these other types of cells here. It looks very complicated. Now, the thing about this, so here we have a spermatogonium, and remember, Stem cells. What are the two main properties? One, stem cells can divide and generate more stem cells. So the spermatogonium are the ones that can reproduce and regenerate so that you can produce more sperm. But the other property of stem cells is that they differentiate, right? So by differentiation, they become different, they become specialized. So once they start to differentiate, they eventually become sperm right here. So again, they can divide to create more spermatogonium, but they can also develop and differentiate to become sperm. So this is what we call spermatogenesis. This process of becoming or starting with the spermatogonium, dividing, and then eventually becoming mature sperm, this is what we call spermatogenesis. And this is what we have spermatids. Actually, I should rewind that, take that back, that stage. Again, sperm mature in the epididymis. So these spermatids, they look like sperm, but they're not quite ready yet. Now, spermatogenesis, this is the overall process. So we're just talking about how you generate sperm from just stem cells. So it's not the same as spermiogenesis. Now, again, very, very similar. And if there's an open-ended question or a fill in the blank, they're not the same. And the thing about spermiogenesis, this is the last maturation state before spermatogenesis. So spermiogenesis is part of spermatogenesis. Spermatogenesis is the overall big process of generating sperm, whereas spermiogenesis is only that last part. Now, this is spermatogenesis. Again, spermatogenesis is referring to the entire process. Spermiogenesis is that last part going from spermatids to form mature spermatozoa. Now, what we have, okay, so a lot of you might not have had genetics yet, but when I'm talking about N, N is a set of chromosomes, so it's like a complete set. So again, remember, in the typical non-sex cell, you have 46 chromosomes, and you get 23 from your mother and 23 from your father. So you have actually what we call 23 pairs. And what we have here, so the thing is that, why am I flipping this around? So to go with that previous figure showing you the development from Spermatogonium to Spermatozoa, if you flip that original figure in your book around, this is showing the development. So you go start with Spermatogonium, you have primary spermatocytes, and then you have secondary spermatocytes, then you have spermatids, and then you have spermatozoa, or sperm. So again, it starts from the outside, then works its way toward the lumen, and then you finally have spermatozoa. Or actually, let's rewind this a bit. So the cool thing about spermatids, so the thing is that they actually have their final genetic material, whereas spermatozoa, they notice that they have not only the head of the sperm, but now they have that tail and mid piece that forms like the motor of the sperm. So the cool thing is that sometimes men who are infertile, they actually are, there's nothing wrong with the genetic material, but something is in their genetics that causes this part right here, the propeller, the head and the mid piece and tail, that actually causes it not to be fully formed or not work properly. So if the sperm can't swim, they can't fertilize an egg. So one type of reproductive therapy is to take the spermatids from these men and inject them into the donor egg or their partner's egg, and then have it fertilized in the test tube or in a petri dish. The cool thing is, like, why? This is already has the final genetic material. It's just that in those men, they have problems forming this full spermatozoon.